The second step in gene expression is the process of translation. So remember, in transcription, we went from DNA to a single-stranded piece of messenger RNA. In translation, we're going to take that messenger RNA and turn it into a protein. So in translation, we are actually going to produce an amino acid chain. This is also called a polypeptide. Now, your book doesn't use that word, but that's the word that I use. So I'm just letting you know, in case I slip up and say amino acid chain, or I'm sorry, and say polypeptide, I actually mean amino acid chain. And then, as discussed earlier, remember that that amino acid chain is going to be folded into a functional protein. So the process of translation requires that messenger RNA that we made in the last step in transcription. It requires a new molecule called transfer RNA, or tRNA. It requires those amino acids that we're going to link together to make a chain, and it requires ribosomes, which are the site of protein production. The process of translation is going to produce an amino acid chain that can then be folded into a protein. Now, this whole process is going to use something called the genetic code, which is essentially the code that is going to translate our language of RNA, our messenger RNA, into the language of amino acids. So we talked about messenger RNA a little bit already. There are a couple of really important RNA molecules in the cell. One is messenger RNA. Now, messenger RNA is a single-stranded RNA molecule. Remember that I said that RNA contains uracil instead of thymine. And then we also have transfer RNA. Now, transfer RNA is also a single-stranded RNA molecule, but it's not linear like messenger RNA. It's actually folded into a 3D shape that gives it a very specific function. So our transfer RNA is going to be the molecule that acts as the code breaker or the interpreter to turn our messenger RNA sequence, or indirectly our DNA sequence for the protein in question, into the language of proteins. So now our book does not draw this as a single-stranded folded messenger RNA molecule. It draws it as this crazy, almost anvil-shaped molecule. And it does that to simplify things and to show you what it's doing. So on one end, we have an amino acid bound. So the transfer RNA is actually going to bring in amino acids to start building our chain. Now, it knows which one to bring in because of an attachment site at the other end that is complementary to a 3-base sequence in the messenger RNA. So every time the messenger RNA says ACG, the complementary sequence UGC is going to recognize it and bring in the correct amino acid. This amino acid happens to be called threonine. That 3-letter code is called a codon. So the 3-letter code is going to signify a very specific amino acid. So I hope you can see how the transfer RNA can actually act as a translator. It reads the nucleotide code and brings in the correct amino acid word, if you will. So the key to the genetic code is a fancy codon table. So a 3-letter code can code for 64 codons. These codons code for 20 different amino acids. So you'll notice that there's more than one codon for some of these amino acids. And that's okay, it just makes things a little bit more convenient for the cell. Also important in this genetic code are the presence of start and stop codes. So remember I said there was that transcription start site? There's also a translation start site, and it is encoded by a single code for an amino acid called methionine. So this AUG word is always going to be present at the start of our proteins. We also have stop codons. So just like transcription had a termination sequence, translation also has a termination sequence. So any one of these three codes, UAA, UAG, or UGA, are going to signal for the ribosome to stop making the protein. And the reason for that is that there's no transfer RNA that carries anything, that there's no transfer RNA that recognizes this sequence. So there's nothing to add when the cell meets, or sorry, when the ribosome sees that sequence on its messenger RNA. So many of these amino acids are coded by multiple codons, and that's okay. The cell will know the proper transfer RNA will bring in the correct amino acid. So for example, there are going to be 4 transfer RNAs that are going to bring in this threonine molecule. So the way you read this is by finding the first position, U, the second position, and then the third position in the middle of the code. So if our code was, let's say, CCG, we're going to start here. We're going to find C. Now we need C in our second row, so that's this box here, and then A. So CCA codes for proline. Let's say we want AGA. So we'll go to row A, column G, third letter A. AGA codes for the amino acid arginine. You do not need to memorize these with the exception of that start codon. So I want you to note that if you see an AUG, that's going to signify the start of your amino acid chain. Now, please keep in mind though that methionine can actually be used inside of the sequence as well. So if you have a start codon for methionine, it's going to be at the beginning of your messenger RNA. If you find it in the middle, it's not another start codon. It's just a methionine that is part of your amino acid code. All right, so let's talk about how translation actually happens. There are 3 main steps to translation. So the first thing we're going to do is we're going to recognize our messenger RNA. The ribosomes are going to bind. We're going to translate. I realize that says transcribe right now. I will fix that in your notes. And then we are going to terminate. Just like in transcription, we're going to stop once we get to the end. So in the first step, we have sort of an initiation step. Now this same figure is found in your book. It's figure 615 with a lot more detail on it. I just took out a lot of the description just because it was really busy for the slide. But if you want to grab that, you can read in detail what it says at each of these places. So in the recognition step, your messenger RNA is going to bind to the ribosome. Now the ribosome is actually made up of 2 parts, the small subunit and the large subunit. Now each of these parts is actually made up of a combination of a third type of RNA called ribosomal RNA or rRNA and proteins. So this structure, this rRNA and protein structure, is the small subunit. Now the small subunit is the one that's going to actually recognize and bind the messenger RNA first. Once that happens, your start site is going to essentially act as a beacon for the first transfer RNA, and it's going to come in with its amino acid and it's going to bind. Excuse me. After that happens, the other half, kind of like the cap or the top to our ribosome, is going to come in and bind. So the large ribosomal subunit is going to bind on top of that. In the next step, we're going to build our amino acid chain. So here was our first methionine. We saw that guy up here. Transfer RNA brought that methionine in. Now the next thing that's going to happen is the ribosome is going to shift a little bit, and that methionine is going to be present at this center location. Now, the next transfer RNA is going to come in to the right-hand side. It's going to recognize the messenger RNA sequence with its complementary base pairs and its correct amino acid. That whole chain is going to be moved over to the right-hand side, and then everything's going to shift. Now, let's pretend it all shifted. Now we've got our growing chain back in the middle. The empty transfer RNA moved to the left side, and then everything's going to shift again. The next amino acid is going to come in. The amino acid sequence is going to be transferred to the right-hand side. Everything's going to shift by 1, and this site in the middle is going to hold our chain again. That last empty transfer RNA is going to be kicked off. The next empty one's going to move over. So just like our our RNA polymerase was like a train on a track, the ribosome acts the same way. It moves along the messenger RNA, reading it as it goes and adding the next amino acid to the chain. Now, if you watch the animations, you really get a better sense of what's happening here. Because this is a fluid process, it's really kind of hard to show without any movement. The third step is termination. So once we get to the stop codon or the stop sequence on our messenger RNA, the same thing's going to happen as in transcription. Everything's going to sort of get jammed up. There's no transfer RNA to add a new amino acid, and so the ribosome is going to fall apart. The messenger RNA is going to come off, and our new protein is now going to be folded and can be used in the cell. This ribosome can go on and bind this messenger RNA again and make another copy if it wants. Or it can go bind another messenger RNA. So this whole process of transcription and translation can actually happen several times. Once the DNA is sort of opened up, another RNA polymerase can come in, make more messenger RNAs. The messenger RNAs can be used for a day or so. Maybe a couple of days depending on the organism before they are degraded until they're needed again, in which case they're built up again. And so when this process happens, we don't just get one protein molecule, but we can actually get multiples. We can get many hundreds of protein molecules every time this particular gene is turned on and expressed. So let's talk about how those genes are turned on and expressed. So our genome is present in every single cell. Now that doesn't mean, though, that every single cell expresses all of the information that it needs. It's kind of like you have all of this information for all of your classes, but you only use biology on the days that we meet, or you only use biology at the times that you're studying for biology. And so the cell sort of acts the same way. It can access the information that it needs through regulation of gene expression. So remember we said that gene expression was just the production of a protein that a particular sequence codes for. Not all cells are going to need the same proteins at the same time, and not all cells are going to need the same protein all the time. So this is how we get different types of cells within our bodies. It's how we account for changes in our bodies at different times in growth and development. So when a baby is in the womb, it's expressing very different proteins than a child or an individual going through puberty or a grown adult. And so there's this process of regulation that allows the genome to be turned on, different parts of the genome to be turned on at different times. So it essentially is controlling whether a gene is being produced, or I'm sorry, a protein is being produced right now. It also controls when it's being produced. Now this is regulated by hormonal cues and all sorts of things that we're not going to get into, but I just want you to understand that the reason that we have different cells and different shaped organisms and things is not only because our genomes differ a little bit, but because gene expression differs a little bit. The environment can also affect the regulation of genes. You know, at different times in your life, you might be exposed to different things, and that will cause different genes to be turned on and turned off. So as I said, changes in gene expression account for differences in cell types and even between individuals. So aside from changes in development or growth, let's look at an example of changes in gene expression. So a 5-year study looked at the health consequences of loneliness. And what they did was look at what sort of genes are expressed in a particular cell in individuals that are lonely and individuals who are not lonely. Now, they use this really cool technique called a microarray, which is essentially a microscope slide to which they have bound messenger RNA. So they have isolated a cell, extracted the messenger RNA, and basically coated this microscope slide with complementary messenger RNA sequences. And so they can see what sort of messenger RNA are being produced at a cell at any given time. So you can imagine this is probably really useful. And so they found that the microarray patterns were different between individuals who were lonely and individuals who said they were not lonely. So in this study, they looked at immune system cells, and they found 209 genes that were expressed differently between lonely individuals and individuals who are not. So this is a fantastic example of how environment can affect gene expression. So they found that these 209 genes were associated with increased gene activity in genes that were related to inflammatory processes. So inflammatory processes are medical conditions that lead to inflammation and swelling and things like that. So they found that there were a variety of diseases that are related to these this difference in gene expression. So some of these inflammatory diseases are arthritis, fibromyalgia, different types of back pain, neck pain, other unexplained swelling. So this is a really cool study that sort of shows us how a cell can change expression in relationship to some sort of environmental factor. And really something that I would not have thought would cause a change in gene expression. It makes perfect sense to me that somebody who is going through puberty needs different genes turned on and off because they're growing and they're changing and their bodies are maturing and things. But the fact that the environment can also affect gene expression is really interesting.